Types of microarray. There are different types of microarray. The most common types, they are DNA microarray, exon arrays, comparative genomic hybridization arrays. DNA microarrays, the examples of the DNA microarrays can be such as cDNA microarray and oligonucleotide microarray and SNP microarrays. Exon microarrays, they usually help to identify the alternative splice site detections. So with the help of exon microarrays, we can identify the alternative splice site variants. Another type of the microarray, which is called as comparative genomic hybridization. In this diagram, we can see that what are the oligonucleotide arrays. In case of oligonucleotide arrays, we have the control cell and the experimental cell. Then the RNA is purified and it is labeled with cyanin green and cyanin red. So in case of control samples, we can see that the spots are visible because here the RNA is labeled with cyanin green and it gives the green color. While in case of the experimental samples that were labeled with cyanin 5 and cyanin 5, it gives the fluorescence of a red color. So in this way, we can identify between the control cells and the experimental cells, because the control cells, they give fluorescence in green color, while the experimental cells, they give fluorescence in a red color. That is due to the cyanin 3 and cyanin 5. In another example, control samples, we have the control samples and the experimental samples. RNAs were purified. They were labeled with the help of cyanin 3 and cyanin 5. Then they are identified in a single plate. We can see here that some of the spots, they are red, and some of the spots, they are green, and some are yellow. So with the help of these green and red and yellow spots, we can check the expression of the genes. Other types of the microarrays, they are called as protein microarrays. In case of protein microarrays, we identify the protein-protein interaction. In case of tissue microarrays, we study about the different gene expression level in different tissues of the organism. Cellular microarrays, which are also called as transfection microarrays, they are also type of the microarrays. Expression microarrays are used to study the expression of the genes. Chemical compound microarrays, in which we identify microarray or we study about the proteomics in case of another type of the microarrays. Carbohydrate arrays, in which we study, which is also called as glycoarrays, in which we study or the amount of the carbohydrates. Another type of the microarray, which are called as resequencing arrays. What is usually required to study the microarray? There are different techniques and procedures which are usually required to study or to perform the microarray. So microarray combine genomics, silicon chip manufacturing, DNA and protein chemistry, signal and image processing, statistics, software skills, and traditional molecular biology experiments. So microarray is the combination of all these techniques. When we have all these techniques, then we can perform the microarray. With the help of microarray, we can study the expression of the genes, or gene expression profiling can be performed with the help of microarrays. On the other hand, microarray also helps to study the single nucleotide polymorphisms. So SNP array, identifying for the single nucleotide polymorphism, are used for identification of SNPs among the population or among the individuals of a specific population. Another type of the microarray is called as methylation microarray. Methylation microarray determines the methylated DNA, mean it determines the difference between the methylated DNA and the non-methylated DNA. Determining which region of DNA are methylated and which region of the DNA are not methylated is usually we do methylation microarrays to check those regions of the genome or of the genes.